Okay, good morning, everyone. So we've been, the last couple lectures, we've been discussing the properties of our cell membranes. And so we've been talking about how the membranes of our cells play a very important role in allowing the cells to be able to carry out all the functions that we associate with them being alive, right? So not only the physical kind of structural support that allows a cell to actually be defined by something like an outer cell membrane, as well as intricate networks of internal membranes that define different types of organelles and different types of areas inside of the cell. And importantly, that membranes are semi-permeable, meaning that membranes are not solid walls. They are actually permeable in the sense that things can move between them from one side to the other side. So this is not only important when you consider that the cell with its cell membrane has to communicate with the outside, and things have to come in and things have to go out, but also that within the cell, the different compartments, like the different organelles, have to be able to serve to concentrate different types of macromolecules and bring them together in order to favor the very specific biochemical pathways that need to happen, right? That would not be able to happen very efficiently if there was no control over how these different types of macromolecules got concentrated and put together. Right? So we looked at some examples last time of the molecules that make up the cell membrane, things like phospholipids, things like glycolipids, things like sterols, like cholesterol. And we appreciated the construction or the structure of the fluid mosaic cell membrane that's made up of a lipid bilayer that's held together by water, right? And having different types of proteins, different types of lipids, and other molecules kind of embedded in it, right? Again, themselves being held in the membrane because of competing water forces, hydrophilic and hydrophobic, right? And how these molecules are charged. So it should come as no surprise to you then, hopefully, right, that it's not enough just to allow things on their own to kind of move in and out of these membranes, right? The very fact that life can exist has to do with the fact that you meaning your cells, are intentionally concentrating things and intentionally moving things across membranes. And so for the next few lectures, we're going to explore this. It doesn't sound like a terribly exciting topic, right? Moving things across membranes, but it's really critical. And I think once we start looking at the different mechanisms that cells use to move things across membranes, I think you'll have a new appreciation for it. So let's start off today talking about membrane transport in the context of simple and facilitated diffusion. So diffusion has to do with the natural flow of some sort of substance across a membrane or through a liquid, having to do just with the natural high to low concentration flow. And I think this is very intuitive, right? Let's say you put a whole bunch of small molecules in water. It's not surprising that over time, those molecules will disperse or spread out through the water, just like putting, for example, the blue dye in a flask of water, as you see here. I think none of us are surprised, right, that at first the blue dye is concentrated where it's being dropped in, but given enough time, right, a few minutes, maybe a couple hours, you come back and you find that you can no longer see the dye being concentrated in one spot. It is naturally diffused throughout the water, and the flask becomes a nice uniform blue color. Okay. You can think about it in terms of this sort of time scale here, time moving from left to right, and the concentration of whatever these molecules are. Let's pretend these are the blue colored dye molecules, right? They go from a high to lower concentration as they spread out, right, until they reach a point of equilibrium, right? Now, why is it that the dye moves out, or why is it these small molecules actually spread out? It might be, on one intuitive level, you think, well, these molecules, these little molecules are bumping into each other, right? And so as a result, they kind of bounce around until they spread out, and that's true to some extent. But again, we forget one of the most important things that's not really pictured very well in the cartoons that we look at when we study cell biology, and that's the presence of water, right? So all of this white space over here in this figure is an abundance of little water molecules that are also bouncing around, right? So just like the blue dye molecules might be bouncing around and knocking into each other, and as a result, just kind of naturally spreading out, so too is this driven by all the little water molecules that are bouncing around, getting between the molecules of the dye. So of course, the water is what we consider a solvent, right? That's the substance into which we are trying to dissolve something. And then the blue dye in this particular example would be the solute, right? And so as the water molecules are bouncing around, they're trying to kind of even out their natural sort of energy of bouncing around. And so as a result, they encourage the solute particles to spread out as well. So this would just be in, let's say just a flask of water like we saw in the photo here. But, this type of property happens, of course, even if you had something like a membrane dividing, right, two different chambers or two different compartments of this flask. So again, if we have all this white space here, not pictured are all the little water molecules that are in here, and we have something like a phospholipid bilayer, right, that's cutting across the middle of this figure here, right, defining two compartments, the compartment on top on one side of the membrane and the compartment on the bottom on the other side of a membrane, right? We can pretend that this is the boundary of a living cell, in which case the bottom part would be all the water that's inside the cell, the cytoplasm, and all the water on the other side would be the extracellular fluid, right? The fluid, whatever that fluid is on the outside of the cell's cell membrane. So here you can imagine that this small molecule, right, will also want to be bouncing around next to each other, and gradually, right, they might make their way through the membrane until you reach an equilibrium where the concentration on either side of the cell membrane is equalized, right? So that would just be the natural kind of tendency if we just allowed this process to happen purely by diffusion. Right? And it assumes a couple of things. One, that there are no necessarily charges involved here, positive and negative charges that might interfere with the way that the molecules move. And also we're assuming that whatever this little blue molecule is, like this dye molecule in our example here, right, that it's not impeded too much by the hydrophobic phospholipid layer, right? Because if this were a molecule that was rejected by the hydrophobic layer, then that would also influence how this diffusion process happens, right? So this is really just a simple illustration of diffusion. So not surprisingly, in a living cell, right, simple diffusion of things across a membrane plays only a small part of the more complicated sort of coordinated effort of moving things back and forth. Because in a living cell, you're not only dealing with the size of molecules and whether or not they're hydrophilic and hydrophobic, you are dealing with charges. Right? And charges can have a big impact on how things move back and forth. So the distribution of charges along or on either side of a cell membrane, right, is not homogeneously distributed. You might intuitively think, well, the actual charge of both sides or either side of the cell membrane maybe is neutral, right? But that's actually not true. It's not true because there tends to be a much higher concentration of proteins, for example, inside of a cell right up against the inner part of the cell membrane compared to maybe right on the other side, the outer side, right? So of course, the peach colored area is the intracellular fluid. The light blue colored fluid on the left is outside of the cell, the extracellular fluid, right? So you have a lot of proteins on the inside, perhaps right up against the inner wall of the cell membrane. So that tends to make the inner wall of the cell membrane more negatively charged. Okay. On the outside, it tends to be more positively charged. Right? The different types of molecules that you're moving back and forth, things like ions, right, themselves carry charges. So you can see there are negatively charged ions like these chloride ions, right? You have positively charged ions like sodium and potassium. And what you notice is that these charges will tend to want to distribute equally across the membrane as well. Right? But because these are different types of ions, chloride, sodium, potassium, the identity of these molecules themselves play a role in how evenly they want to distribute across. Remember, if it was just left up to simple diffusion, right, the concentration of sodium ions on one side would eventually even out with the concentration of sodium ions on the other side, right? If it was just a matter of simple diffusion of sodium. But in reality, look what happens in this cartoon here. You have an uneven distribution of these ions. You end up with way more sodium ions on the outside of the cell. Right? That's why this triangle here is drawn this way. Higher concentration of sodium on the outside, lower concentration of sodium on the inside, higher concentration of chloride ions on the outside versus lower on the inside, right? And higher concentration of potassium ions on the inside of the cell compared to the outside, right? So clearly, the movement of these ions is not left up to them and simple diffusion, right? Clearly, the cell is somehow involved in, on purpose, separating out, right, where the potassium ions go, where the sodium ions go, all right? And that creates then an electrochemical gradient, right? In other words, a gradient, when we say gradient, what do we mean? A gradient is like, the ramping up of something is like a gradient from low to high, right? So you can think about that in terms of concentrations, right? Low to high concentrations form a gradient, right? So clearly, the cells are dealing with not only the chemicals that need to be moved back and forth, but the electrical charges of those chemicals. So that's why we say an electrochemical gradient is actually at work here. All right. So this is an overview of many of the types of membranes that we will be looking at, right, that all make up a living cell. So we've started looking at this already. Now, don't let this overwhelm you, right? It's not like I'm asking you to memorize this whole thing here, because the point is, this is more about just giving you examples of different things we're going to study. So, for example, the outer cell membrane, we'll take a look at today, examples of how there are certain proteins, like this little purple dot here, that play a role in controlling the diffusion of certain types of molecules, right? As well as playing a role in actively moving certain molecules. We've already looked at the nucleus before in the context of understanding how different things move in and out of the nucleus. How, for example, you imagine that you have a concentration of nucleic acids in the nucleus, like deoxynucleotides, right? Your DNA would be concentrated in the nucleus. You would have nucleotides coming into the nucleus in order to help assemble things like DNA. You would have mRNAs moving out of the nucleus, right? As well as certain proteins moving in and out. So clearly, the nuclear membrane or the nuclear envelope must have different types of things like proteins, like these little pink little donut shapes that are involved in moving things in and out, right, to keep that under control. We will look at later on when we examine cellular respiration, the role of the mitochondria, right? The mitochondria itself is very complex. Not only does it have its own membrane on the outside, but inside the mitochondria is an extensive network of little membranes, right? And it's across these membranes that things like protons get moved. Protons that get moved across the membranes and then are allowed to flow back through those membranes through special proteins that end up driving synthesis of things like ATP, adenosine triphosphate, which is used by the cell as its energy currency, right? In other words, the energy molecule that helps distribute energy throughout the cell. We will learn about that in a subsequent lecture. And this particular cell has this big green shaped structure here. This is a chloroplast, right? So a chloroplast is found in plants. So this cartoon is a plant cell, right? The cells in your body don't have chloroplasts, but this is where the chlorophyll is, the chlorophyll that can perform photosynthesis. And again, we'll study that later on in the semester as well, right? So those will all be more complicated examples of how membranes are used to drive important functions. But today we can just kind of focus on the outer cell membrane, right? So I said we'll look at different proteins like this carrier protein up here that can help move glucose through the membrane. We'll look at examples of this one here. This is aquaporin that's in charge of moving water in and out. And there are different types of proteins that act as pumps that can actually move ions across in different directions. And importantly, I don't want to forget to mention, if you look over here, this is oxygen and carbon dioxide, right? Notice how there's no proteins in the cell membrane drawn here because gases like oxygen and carbon dioxide are free to just diffuse, just based upon concentration gradients. All right, let's take a look at this. So molecules that are going to be moved across a membrane in the cell can either be moved along a concentration gradient, meaning the natural tendency for molecules that are in a high concentration on one side, right, to want to kind of distribute and move to a lower concentration on the inside to sort of even out the concentrations on either side of the membrane, right? Anything that just relies upon the gradient alone to kind of move things is a passive process, right? There's no energy required in that because molecules, the solute dissolved in water, right, will naturally want to move from a high to low concentration. So moving along a gradient, we can call that a passive process. You can also say it moves with the gradient, right? And I think actually the slide that you guys have in the PDF form, I called it moving with the gradient, right? Same thing, but maybe along is a better way of illustrating that it's going from high to low. So for example, an example of passive diffusion or simple diffusion is if you just have, like in this cartoon on the left here, these small little blue molecules, whatever these molecules are, right? They are dissolved in the water, they are the solute, right? And in this case, if they can just kind of flow through the phospholipid bilayer, going from the high concentration on one side to the low concentration of the other side to eventually, kind of even things out, that's just simple diffusion. That's about as simple as it gets, right? So in this case, these small molecules flow readily through the membrane because they are small, right? They're small enough to fit very easily between the phospholipids that make up the membrane. And maybe they are, for example, hydrophobic, right? If they're hydrophobic, that means not only are the water molecules on the upper part here, just bumping into them and shoving them all over the place, including shoving them towards the phospholipids over here. But because they're hydrophobic, they very easily move through the membrane, right? And then would end up on the other side. Okay. But if those solute molecules are maybe bigger, for example, here, maybe we have a simple sugar, right? They might be too big to actually move easily through the cell membrane by themselves. In that case, you could have special proteins embedded in the membrane, like this big green blob here, right? Remember, this might be a protein that is one long polypeptide chain, right? Because it's a protein, but maybe it zigzags back and forth through the membrane multiple times until it sort of comes together with some sort of little channel through the middle. Right? And maybe the shape of this protein, like maybe in this area here, is just the right shape because of its amino acid sequence and all the little side groups of the amino acids, right? To be able to bind something like this simple sugar, right? And therefore facilitate that sugar's movement to the other side of the membrane. So again, what's the driving force here that's driving these sort of red molecules from the top to the bottom, right? It is still the gradient, right? They naturally want to just flow through to the lower concentration side, right? So therefore, does this protein actually need to actively do anything? No, right? The force is just provided by the high to low concentration gradient. So that's why we say this protein is just facilitating or helping this along. So again, it's a passive process and it's called facilitated diffusion. On the other hand, there most certainly are going to be examples where you need to move things in the opposite direction against the concentration gradient. Right? So in this case here, you have these purple squares, whatever they are, right? And they are at a pretty low concentration on this side of the cell membrane, but the cell needs or wants to be able to move as much of these across and into the cell as possible, right? Because this might be something the cell really needs to have in a high concentration on the inside. But the direction of the normal gradient of concentration, right, would not favor this. And I think that's kind of obvious, right? The direction of this big purple arrow, if it was just left up to concentration gradients, would be flowing the other direction, right? So in order for the cell to concentrate things inside, right, this green protein blob here cannot just be passively, right, involved. It actually has to actively participate in moving these things through. Right? So the way it's drawn here, imagine if this purple square binds on this side, right? This protein, the big green blob, has to actually change its shape, right, to sort of force the purple square out the other side. Right? But the protein doesn't want to just passively change its shape, because if it's open to the inside, like you see here, right, you have all these purple squares on the inside that they would preferentially, right, want to shove the protein in the other direction. Right? So in order for the protein to be able to kind of force the purple squares through, it requires energy, right, in the form of, for example, a molecule of ATP. Right? A molecule of ATP that has 3 phosphate groups on it. Those phosphate groups represent high energy bonds, right? So if this protein were able to break one of those phosphates off, break that high energy phosphate bond, right, and then have that energy used to maybe bind onto the protein and change the protein's shape, that amount of energy, right, of forcing the protein to change its shape into the shape that you see on the picture here, right, would be a stronger energy than any of these purple squares on the inside of the cell being able to counteract that, right? So moving things against a gradient will require energy, and we call that active transport. Today, we'll focus mainly on examples of diffusion, simple and facilitated. And then in the next lecture, we'll look at some examples of active transport. But hopefully you already appreciate that all 3 types of moving things across membranes is very important. All right, so let's focus right now on comparing simple diffusion with facilitated diffusion. Right? I said that simple diffusion is just when things can, because they're small and maybe hydrophobic, just flow through the membrane purely based on their concentration gradient. Right? So here's an example of simple diffusion. Right? These little red molecules, this solute, whatever it is, right, can just easily make its way across the phospholipid bilayer. So in this figure here, we're saying from left to right on the x-axis is change in the solute concentration. In other words, at this point over here, let's say you have no solute, right? None of these little red molecules present. And then as you move more towards the right, you're increasing the amount of the red molecules on the outside of the membrane, the peach-colored side of the membrane. What would happen? I think you can kind of intuitively see that the more concentration on the outside you have of the little red molecules, right? The y-axis is the rate of diffusion, right? Because these are just allowed to flow through, the more you have on the outside, right, the faster and more of the molecules flow in, right? Purely just based on the concentration of these red molecules on the outside. So it's linear, right? Because there's nothing to actually stop them from moving through. So that would be simple diffusion. A simple diffusion follows a linear kinetics of rate of diffusion. However, what if the molecule was bigger, right? For example, like a sugar, right? In this case, these green molecules cannot get through the membrane on their own, right? They cannot wiggle their way through the phospholipid bilayer. So you need to have some sort of proteins involved, right? Like these proteins, the blue one here, right, are helping or facilitating the green molecules to go through. But again, it's all being driven by concentration gradient. But now, right, you get sort of a bottleneck. So let's see how that affects this kinetics. So if you start off with very little of the green spheres here, as you increase those, right, on the surface of the cell, let's imagine you have a ton of these blue protein channels ready to help move the green balls through, right? So as you start having more and more of these green molecules, there are plenty of these blue molecules to let them through. So as a result, right, it's not a linear, well, it is a linear response at first, but the slope is much steeper, right? Because as these green balls arrive on the surface, right, there are plenty of available, what was I going to call it? The blue proteins to let them through. But what happens once you increase the concentration of these green solutes? At a certain point, right, you have way too many of them on the outside. The concentration is going up and up, and you begin to get a bottleneck, right? Because there's only a finite number of these blue channel proteins on the surface, right? Only a finite number of places where the green molecules can come in. So you begin to saturate, right, the transport, right? Because there's only a maximum number of these blue molecules available to let these green things through, right? So at a certain point, right, you end up getting this bottleneck kind of effect. So that's just like, if we're talking about fans wanting to get into a sporting event, right? So here's the Stan Sheriff Center, which very sadly over the last 18 months or so has kind of looked like this on the left, right? But think about it this way. The gates or the doorways into the Stan Sheriff Center are like these blue channel proteins, right? They are the way in for the fans to get in, right? So once you start having some fans show up, right, the rate at which they can get in at first is a pretty steep linear relationship because there's plenty of doors. Nothing is sort of stopping or bottlenecking the fans from getting in, right? However, if you start having too many fans outside, right, the number of entrances remains fixed or the same. So therefore you begin to saturate these entrance gates, right? And you end up getting a buildup of solute on the outside or fans on the outside, just waiting to get in. And the overall kinetics would actually plateau, right? Once you've maximized how quickly people can pass through these gates, right? That would be a leveling off of the maximum rate at which people can get in, right? So that would be an example of facilitated diffusion of things moving into a cell. So why is that important? Because since facilitated diffusion, right, depends upon different things like proteins on the membrane to actually allow things to come in, right? This actually provides an opportunity not only for the cell to control the rate at which things are coming in, right? Because after all, the cell can control how much or how many of these channel proteins it has on its surface. Remember, these channel proteins are all expressed originally by whatever genes code for the proteins that make these channel proteins, right? So a cell can regulate the expression of those genes, thereby controlling how many of these finished sort of gateway proteins are made to put on the surface, right? Also, this provides very interesting clinical, therapeutic, biomedical opportunities for modulating the rate at which cells take things up. Because let's imagine, for example, in this cartoon here, right, you have this protein on the cell surface that transports or is involved in the facilitated diffusion of a particular substrate, this light blue colored molecule, from one side of the cell into the cell, right? So the rate of the reaction would be the transport rate across this membrane. Right? If all we had in this picture was that purple protein, the transport channel, and the substrate that's moving through, then on the right, you would see this purple line, right? This is the purple line that we saw on the previous graph, even though I don't remember what color it was on the previous graph, but it's the same thing, right? You have a finite number of these purple proteins available on the outside of the cell at a given time. So as you increase the amount of substrate or solute on the outside of the cell, at first, right, there are a ton of available purple gates to let the solute in. So the slope of the rate of diffusion is very steep, but at a certain point, you begin to max out the number of entrances, right? And so even though you keep increasing the substrate concentration on the outside, you plateau. Right? So that part's exactly the same as that example with the Stan Sheriff Center and the entrance gates for the fans, right? But look, it could be that this particular protein not only is the right shape to bind this light blue substrate, but maybe, right, you could have some other molecule that also binds to that protein. I say also, not in the sense that it's happening at the same time, but you can see in this cartoon here, if this blue competitive inhibiting protein binds to the protein instead, right, that means the desired light blue substrate cannot bind. So therefore, this molecule, this dark blue molecule, whatever it is, would be acting as an inhibitor, right? Because it's inhibiting the movement of the desired substrate into the cell, right? And this would be functioning as a competitive inhibitor, right? What does that mean? Because the protein can bind either of these two at the same time, right? So they would have to take turns. If one is bound, then the other thing is not bound, and vice versa. So it's like there's a competition for binding to the purple transport protein, and that would be the green line over here, right? So what the green line is showing is, let's say you have now this dark blue competitive inhibitor present, and you're not going to change the concentration of it, right? It's just there at the same concentration throughout this whole graph, even though you're still increasing the concentration of the light blue substrate that you want to move, or that the cell wants to move in there. So what you would see is from the very start, right, the rate of reaction of being able to move the light blue molecule is already inhibited, right, because you have this dark blue competitive molecule competing for the same binding site on the protein, right? But because this is competitive, as you increase the amount of the desired substrate, and this gets higher and higher while keeping the concentration of the competitive inhibitor the same, eventually you end up out-competing the inhibitor, right? So eventually you're able to essentially join up again with the maximum rate of diffusion or rate of reaction. All right. Another scenario would be, what if the inhibitor molecule doesn't actually bind to the same surface that the substrate or the light blue solute binds to, right? Instead, this molecule, this orange molecule, binds somewhere else on the transport protein. But because it binds somewhere else, it still changes the shape of that transport protein. And then this upper binding surface up here is no longer the ideal shape to be able to bind to the substrate over here, right? So this blue thing, if it came down, yeah, it would still fit there, but you can see this part of the transport protein has changed its shape, so it no longer stably hangs onto the substrate, right? And thereby, this non-competitive inhibitor would still be getting in the way of the cell being able to move the light blue molecule in, right? So that would be this blue line over here, right? If you have a non-competitive inhibitor held at a constant concentration, right, even if you're trying to increase the amount of the substrate that's being moved through, because the non-competitive inhibitor, right, simply changes the shape of the molecule, at a certain point, you see it plateaus out, but you've dramatically decreased the overall transport rate. So why is this important? Because if you're talking about designing pharmaceutical drugs, medications that work by, let's say, preventing cells from moving certain molecules into them, right? So for example, things like, certain types of molecules that might be involved in neurotransmitters being sent between different neurons, right? A competitive inhibitor, right, would still work, but the problem is, right, because it's competing against the actual substrate, right, at a certain point, it would lose its efficacy if you have a lot of that substrate available. However, a non-competitive inhibitor, right, is able to prevent transport, right, potentially at much lower concentrations than a competitive inhibitor. And the best part is, right, it doesn't matter how much of the substrate is present, having this non-competitive inhibitor can be more effective. And so for therapeutic drugs, right, there's always this goal of designing non-competitive inhibitors, because that way you can get away with giving the patient much lower doses of the drug and having very strong efficacy, right, or effectiveness. All right, so let's kind of review here. We've been talking about gradients playing a role, right, in determining how things are transported across a membrane. So if things simply diffuse down a gradient from high to low, right, this would be, for example, if you put a tea bag in hot water, many of the molecules like tannins and other things that, have the flavor of the tea, they simply diffuse out of the tea bag into your tea, right? And that's just simple diffusion. And the tea bag is just a membrane that is permeable or semi-permeable here, because it allows the small molecules, the tannins that give the tea the color, and the flavoring molecules, those can move through the paper filter bag, right? But large things like the actual leaves of the tea cannot move through, right? So that's a perfect example of a semi-permeable membrane, right, that's functioning through simple diffusion. Right. If you had little holes or little pukas poked in the tea bag, you accidentally tear it, then you know what happens, right? Then you'd have tea leaves escaping through the little hole, right? In which case that might be more like facilitated diffusion, right? The tea leaves would love to get out of the bag, but they're limited how quickly they can get out of the bag by the size of the little tear in the tea bag. Okay? So the opposite would be moving things against the gradient. Right? So that would be, if you imagine being able to collect all the little molecules that have diffused out into the water here and somehow getting them back into the tea bag. I don't think it takes a lot of imagination to know that that would require a lot of effort, right? So moving things up a gradient would require energy, right? And that'd be active. So this table below helps compare and contrast simple diffusion, facilitated diffusion, and then active transport, which again, we'll talk about active transport next lecture. And it gives you an overview of different types of ways of thinking about them in terms of what types of solutes might be transported in living cells according to diffusion or active transport, the thermodynamic properties involved, and also the kinetic properties, right? That's what we've been talking about with those curves. So let's look, simple diffusion. Right? So the types of solutes that can just flow through a membrane through simple diffusion are small polar molecules, things like H2O or glycerol can move through. Small nonpolar molecules like gases like oxygen and CO2 can just simply diffuse back and forth, purely based on concentration gradient. And some larger molecules, as long as they're nonpolar, so in other words, they tend to be hydrophobic, can also move through the cell membrane just by simple diffusion. So things like oils, right? So fatty acids, not surprisingly, fatty acids can move through the phospholipid bilayer because after all, the phospholipid bilayer is just made up of fatty acids, right? So short fatty acids can move through. Steroids, right? Things that are based around cholesterol, they can also move through as well. So thermodynamically, the direction relative to the electrochemical gradient is they're moving downhill, right? From high to low. Is there energy required? No. Is there an intrinsic directionality? What does that mean? That means if you just have a phospholipid bilayer, right? Is there something that actually says right off the bat that things have to move one direction or the other? No, there isn't, right? This, in case of simple diffusion, right, that could change. The direction of the arrow could change purely based upon concentration gradient. So therefore, there is no intrinsic directionality to this at all. And then the kinetic properties, right? Is there a membrane protein required to accomplish this? No. Can you saturate this? No, simple diffusion won't saturate, right? Because there's nothing that actually bottlenecks the reaction. And is there an opportunity for any kind of inhibition, like competitive inhibition? No, right? Facilitated diffusion, the types of things that are moved would be small polar molecules again. So as we will learn, water, glycerol, they can move through the membrane just by diffusion, right? But the membrane does represent a barrier, right? So it's not like they're flowing through as though there's no membrane there. Right? So there, not surprisingly, will be certain proteins that actually help to facilitate the rate at which these molecules can move through, depending on what the cell needs. Large polar molecules like glucose need facilitated diffusion to get through, and ions, right? Charged particles like sodium, potassium, and calcium need to actually have facilitated diffusion, move them across. Okay? Again, thermodynamically, it's still being driven just by the concentration gradient, right? This is still diffusion. And likewise, no energy is required, and there's no intrinsic directionality to this. Kinetic properties, there needs to be some sort of membrane protein involved in order to facilitate the movement of these particles through. Can you reach saturation? Yes, right? Because anytime when you need to have proteins to move things through, you only have a finite number of those proteins, right? So they sort of represent the bottleneck for how fast you can actually move things in. And is there an opportunity for inhibition? Yes, right? Like the example we looked at when we were talking about the design of medications and drugs, right? You have the opportunity, if you can somehow change the shape of these proteins by binding something else to them, right? You can affect whether the cell can successfully move things in. All right. So active transport, right, which we will introduce today, but we won't talk about until next time. Active transport moves certain large polar molecules through, right? So I know this is confusing, but there are certain cells in your body that need to use active transport to move glucose in. Why would you need to do that? Well, imagine there are certain cells of your body, right, that it's not enough to just wait for the concentration of the glucose outside to be higher than the inside in order to move things in. Maybe those cells already have a ton of glucose in them already, and yet they still need to pull more glucose in. What kinds of cells would those be? Well, the cells of your liver, right? Because the job of your liver is to collect all the extra glucose that's floating around in your blood after you have a big meal, bring it into the cells of the liver and turn them into glycogen, right? So inside those cells, there's already a ton of glucose, right? Much higher than the surrounding fluid of those cells. And yet the cells still need to move more glucose in. So in those cases, glucose needs to be moved through active transport. Thermodynamic properties. So we are now saying active means you're moving things up against the normal concentration gradient. This would now require energy. And would there be an intrinsic directionality? Yes. That means these proteins can only work in one direction because the way that they change their shape is to intentionally, right, shove molecules from one side to the other against the gradient. Right? Kinetic properties, are there membrane proteins required? Yes. Right? Saturation kinetics, yes. Competitive inhibition, yes. There's the opportunity for those as well. All right. So talk your way through this table, and it'll help you understand the content of this lecture and the next. Let's look at some examples. Simple diffusion, right? Gases, as I already mentioned, just simply diffuse back and forth through there. So if you look at your lungs and the little alveoli or the little pockets in your lungs where the gas exchange takes place from the air that you breathe in and then the gases that are in your blood, right? So on the bottom here, here's a capillary, a small blood vessel flowing right up against one of these little alveoli, little sacs, right? And you can see quite literally the red blood cells that are flowing by here, carbon dioxide diffuses out of those red blood cells, right, into the air inside the alveoli, and oxygen that's in the alveoli diffuses the other direction into the red blood cells, purely based on concentrations of higher to lower CO2, higher to lower oxygen. Facilitated diffusion, looking at something like glucose. Here we have this protein on a red blood cell. This is a glucose transporter known as GLUT1, right? So GLUT1 is a carrier that transports glucose into the red blood cells, right? From a high glucose concentration on the outside to a low glucose concentration on the inside, right? This is how our cells can actually take up glucose in a passive way. Here is the structure of the GLUT1. So you can see here, this is the crisscrossing polypeptide chain that makes up the GLUT1. These green things are the phospholipid bilayer. And then you have the water that's on the outside and the water that's on the inside. I like this figure because this one, they actually bother to show you all the little water molecules, right? And so the glucose will bind to the GLUT1 transporter, right, the carrier, and it'll be moved across. Let's look at how this happens. This all has to do with the shape of the GLUT1 protein changing its shape depending on how glucose is bound to it. So on the outside of the cell, in the cell membrane, you have this glucose transporter, this GLUT1 protein. It starts off maybe with it sort of being open to the outside of the cell. We'll just call this a T1 conformation. Right? On the outside, you have a lot of glucose, and the glucose binds to the GLUT1 transporter because the GLUT1 transporter is just the right shape to bind it. Right? When it binds, you can see here, you get a T2 conformation. The GLUT1 transporter protein changes its shape because now suddenly it has this glucose molecule in it. So it naturally changes its shape. So it opens to the inside of the cell. Therefore, the glucose molecule, because remember, this glucose molecule is not covalently bound here. It literally is just sitting here because of shape. So when it opens to the inside of the cell, there's the likelihood that it can fall off. And then it's inside, right? The glucose has now been moved to the inside. And then the GLUT1 transporter then, without the glucose in it, resumes its T1 conformation. This is why it's a passive process, right? Because just as long as there's higher numbers of glucose on the outside compared to the relatively lower numbers of glucose on the inside, right? That means the glucose will tend to bind to the outside more, right? And change the GLUT1 transporter shape and send the glucose in, right? If the concentrations of glucose were the same outside as inside, then you would imagine that on the inside, you'd have just as much likelihood of a glucose molecule binding, right? And then this thing might, change its shape or actually force the GLUT1 transporter to change its shape in the other direction, right? And move the glucose out. So it's all just driven by concentrations. So these types of carrier proteins can move things in pretty fancy ways. So for example, they might be designed just to move one molecule. So here you have a solute, whatever this S is, moving through a carrier. And if that's all that this carrier protein does, like the GLUT1 transporter on the previous slide, then it's a uniporter, right? It just moves one thing through. Right? However, things get a little bit more intricate and clever. There's nothing to say that this particular protein carrier molecule, whatever it is, might be shaped in a way that it can actually bind two different solutes at the same time, like solute A in red and solute B in blue, right? And once those two things bind, right, then both of those molecules are moved in at the same time. That would be a situation where, let's say, the cell needs to have relatively equal amounts of solute A and solute B at the same time, so it would make sense to move one molecule of each. So that would be a symporter, right? And some of these molecules, these proteins, are even more clever. Let's say this particular purple transport protein over here, it binds solute A on the outside, and when it changes its shape, it dumps solute A into the cell. But as it's open to the inside of the cell, that's the right shape to bind solute B that's been floating around inside of the cell, right? And that causes the transporter to move in the other direction, and it ends up kicking solute B out, right? Again, the red and blue arrows are just driven by the different concentrations of these solutes, right? But it all has to do with this protein being the right shape in order to bind things, change its shape, and cause things to happen. Okay, I know we're a little bit over time, so I'm just going to finish with this example and call it a day. I mentioned how water can diffuse through the cell membrane, right? But the reality is the membrane still is a barrier, right? So what if you needed to move a lot more water through than simple diffusion would allow? Well, our cells do have carrier proteins on their surface that are designed just to allow water to move through. Right? So these channel proteins are known as aquaporins. So you can see here in gray is the shape of this aquaporin protein, right? So it'd be going through the membranes. You don't see the membrane here, but this would be extracellular space on the one side, and then the inner cytoplasmic space on the other side. And these red things are water molecules, right? I mentioned this to you in the last lecture, but these aquaporins are amazing. They are shaped exactly right so that the water molecules, as they're flowing, right, from high concentration to low, right, have to move single file through the protein, right? So you get very fine control over water movement. Okay. So what types of cells might need to have heavy dependence on these aquaporins in order to move a ton of water through them? What kind of cells in our body handle a lot of water? Well, kidney cells. Right? The cells in our kidney that filter our blood and need to be able to sort of recover the water from our blood, because you don't want to lose a lot of water as the blood is being filtered. Right? These cells, as you see pictured here, are the cells of your nephrons in your kidneys. And so this particular structure here is inside of a nephron. And what they're staining for in brown is which ones of these cells have a lot of aquaporin proteins on their surface. Right? And you can see a lot of these cells in these groups here, right, have a lot of aquaporin proteins on the surface because they're moving a ton of water in and out of them as part of the filtering. Right? How much water is actually handled by your kidney cells each day? Have you guys thought about this? Well, a typical person has somewhere between 4 to 6 liters of blood in your body, right? So you figure, well, yeah, they probably filter all of our blood, right, in one day to keep it clean. How many liters of water do you think these cells of your kidneys actually handle? Right? It's an astounding amount. Right? Our kidneys process around 180 liters of water every day. Right? That's way more than just simply the fact that you have 4 to 6 liters of blood. Right? And you say, well, I don't get that. Right? Because that's way more than our total blood. Yeah, it's because they're constantly going, right? So it filters the blood over and over and over again, right? So that's the equivalent of 90 2-liter bottles of soda that your kidneys are filtering every single day. So that means the cells of your kidneys, right, have a ton of aquaporin water channels proteins on their surface, way more than other cells do. right? How do they end up with way more? This goes back to gene regulation, right? Because every cell in your body that has a nucleus has the genes that code for this aquaporin proteins, right? It's just in most cells in your body, right, the gene is expressed, so you have a moderate number of these water channels on the surface of the cell. But in specialized cells that are meant to handle a ton of water, like those kidney nephron cells, right, they have very high elevated expression of those genes that code for the aquaporin proteins, right? So they have a ton of the aquaporin proteins on their surface, right? So excellent example of how gene regulation can make things like a kidney nephron cell behave very differently than some other type of cell in your body. Even though all the cells have the gene to make the aquaporin, the kidney cells go nuts, right? And make a ton of the aquaporins so that they can move this much water. Okay. All right. Okay. Let's go ahead and move to the chat, and I'm happy to answer any questions you have in the chat. There's a question about something acting hydrophobically on the inside of the cell. I'm not sure what that is. Maybe if you want to type that again, I can answer that. So I'm still a bit confused on why the initial rate of diffusion is so much higher for facilitated diffusion versus simple. Is it easier for molecules to pass through the channel proteins just compared to just through the membrane until the saturation point? Yes, that's exactly it, right? Because imagine the Stan Sheriff Center, right? And imagine you had no gates at all. Well, that wouldn't be a great example because then people couldn't get in at all. But imagine, right, that you have a cell membrane where you have something that can move through on its own, but the fact that there still is a membrane there, it is a barrier, right? It's not going to move from point A to point B as quickly because there is a barrier there, right? So if you have openings in that barrier, like carrier proteins, right, the rate at which these things can move in is much faster at the start until you have so much solute on the outside that you end up getting that bottleneck happening, right? And you reach the maximum diffusion rate based upon how many of these entrances that you actually have for the solute to actually go in. But yes, you understand it correctly. All right. What would be an example of something having intrinsic directionality, right? I'm still unsure because doesn't diffusion naturally go in one direction? So hopefully, I know you probably asked that while I was just giving some examples of diffusion, but now that we talked about active transport, right? Diffusion itself has no intrinsic directionality. If you have a membrane and the concentration above the membrane is high and the concentration below the membrane is low, then yes, it'll move from top to bottom, right? But if you changed it around and suddenly the concentration on the bottom was high and the concentration on the top was low, things would move the opposite direction, even though the membrane hasn't changed at all. That's what we're saying. The membrane itself doesn't have any intrinsic directionality of which way it's going to let things diffuse, right? But once you talk about active transport and you actually have proteins that are specially designed, right, to take things from a low concentration and shove them in the opposite direction into a high concentration, right? Those, you can imagine, those proteins are very specific shape, so they can only do that in one direction. So therefore, active transport has an intrinsic direction determined by, the orientation or the shape of the proteins that are involved. Okay. All right. All right, good. I'm glad. All right. So I hope you guys have a great rest of your Monday. And I'll see you on Wednesday. All right. All right. Bye-bye.